today is Dr. Mac Burnham, who is an epilepsy researcher and professor of pharmacology at the University of Toronto. Dr. Burnham also serves as co-director of Eplink, an epilepsy research program. Ewan and I got to take his neuropsychopharmacology course last semester, where we studied the neurological phenomena of pain, anesthesia, seizures, and more. So thank you so much for joining us today, Dr. Burnham.
Glad to.
To start off, we learned about some of your past research in PCL 475, some of your research on anti-seizure drugs especially. You also mentioned that some of your current work focuses on studying new anticonvulsant drugs. So we were wondering about what it is that you're studying currently at your lab.
We're very interested in finding new ways to stop seizures. With the present drugs we have, and there are more than 20 on the market now, we only get control in about 2/3 of the patients with seizures. So the other third go on having seizures. And if you have uncontrolled seizures, you have a very poor quality of life. It's very difficult to live with uncontrolled seizures. The particular type of seizures that we focused on are called complex partial, or now that they've renamed them, we're supposed to call them focal impaired awareness seizures. These are the ones where people don't collapse, don't convulse. They just stop what they're doing and sort of stare blankly. They look like absence seizures in children, but they last much longer, and the EEG pattern is different. These complex partial seizures, or FIAS, are the most common seizure type in adults, and they usually resist drug control. So probably the majority of people that can't get their seizures under control have complex partial seizures. Beyond that, the complex partial seizures are particularly associated with some of the psychiatric comorbidities of epilepsy. So you see more depression, more anxiety in people with this seizure type. So we really, really want to see if we can find something to control it. Some years ago, we evolved an animal model of seizures called the amygdala kindling model. And we've been looking for years for a drug which would suppress the particularly focal aspect of the amygdala kindled seizure, which is what we think is a good analog of the human complex partial seizure. These days, we're working with the cannabinoids, cannabis extracts, CBD and THC. Our major interest is in CBD, of course, because it doesn't make you high. But both CBD and THC will stop seizures. It's been shown in animal seizure models, and it's been shown in our model. And I think particularly interesting in our model is that these drugs will suppress the amygdala focal seizure, that component of the generalized or of the kindled seizure, which we think is a good analog for human complex partial. So we're interested in taking it into clinical trials, and that's what we're working on next. We're going to work with Dr. Peter Carlin over at the Toronto Western Hospital and see if we can use the cannabinoids to treat people, humans with complex partial seizures.
So for those of us who are probably not as familiar with the mechanisms of epilepsy, can you explain what the amygdala kindling model is?
Yeah, it's very simple. People with complex partial seizures, and I'm going to use the old term because I think people understand it better, usually have a focal seizure type. So the onset is localized, and it's very often in the temporal lobes, which in human being means where the limbic system is. And very often it is the amygdala, the hippocampus, or the adjacent piriform cortex that originates seizure activity. As long as it's localized, the person is still conscious and in contact with his or her environment. And what they will report is a strange feeling of fear, or a feeling of bad smell, bad taste. Those would all be common. And then we think that the seizure activity spreads partially to the rest of the brain, and we're not sure where it spreads to exactly. But that's when the person suddenly loses contact. They often look like they're actually hallucinating to me, but we never know because they never remember after the seizure what was going on in their mind, and they don't remember the seizure period at all. Basically, in our animal model, we implant electrodes into the rat's head. We let the rat heal up for a couple of weeks, and then we begin to trigger little local after discharges in the rat's amygdala, one of the limbic structures, or what would be called in a human a temporal lobe structure. And the rat seems fine at first, but after a while, you begin to see that the seizure discharge is spreading more and more from the focus. And at that point, the rat begins to stop what he's doing and stop and stare, which is really a bit like a complex partial seizure in humans. And then if you go on long enough, the animal will start to actually convulse. So there'll be the focal seizure, the partial generalized, and then the fully generalized seizure with a convulsion. And of course, people with complex partial seizures also can go on to secondarily generalized convulsions. So that's it. We're basically triggering a limbic seizure, and we're looking for drugs to try to stop that limbic seizure. If we could stop the focal seizure, we believe we would be able to stop the whole partial generalized and convulsive seizure as well. We have been working with both CBD and THC in our lab, and this is something a lot of labs haven't done. So this working with a combination is something that's particular to us. Now, CBD and THC have completely different mechanisms of action, although they both stop seizures in different ways. And they often occur together. If you get cannabis extracts, they often have some CBD and some THC in them. So we wondered what the combination would be like. And what we're finding is that the combination works better than either drug alone. And so we're interested in going into our clinical trials with a mixture of mostly CBD, but with a little THC as well.
You mentioned that CBD has mostly an effect on the amygdala. Would that mean that you're aware of this fact because you tried it on the focal onset seizures only?
Well, we've tried it essentially mostly with focal onset seizures. What we find is that first it suppresses the generalized seizure, and then at higher doses, it suppresses the focal seizure. But by the time you can suppress the focal seizure, you're still not seeing toxicity. CDB is really very benign compared to a lot of the anticonvulsant drugs that are out there these days. So not only might it have the advantage if it might stop this particular seizure type, but also the side effects wouldn't be too bad. THC we would use only in small doses because, of course, the side effects with THC, well, that's what people take it for. They take it to get high. We've got enough drugs that have sedating side effects. We don't need a new one. But we're hoping that we could use mostly CBD with just a little THC to maybe get some synergy.
So how was it that researchers, I don't know if it was your lab or maybe another lab, first discovered these anti-seizure properties of CBD and
Oh,
THC?
they have been known since antiquity. By the time you get into ancient Indian medicine, you're already seeing it used for seizure control. And cannabis was used for seizure control all the way up until the early 20th century. The problems with using cannabis itself is that the strength varies from batch to batch and that the shelf life is not very good. So when they began to get the more modern anticonvulsants, cannabis went out as a treatment for epilepsy. But it came back really, it was brought back by the families of people with kids with seizures, because what they found sort of word of mouth was that cannabis could stop seizures that would resist all the other anticonvulsant drugs. This was first discovered by a family really in Colorado. And after that, it spread through the community. And so by the 70s, people were really seriously looking at the cannabinoids as anticonvulsants, particularly a group in Utah that did a lot of experiments with them in the 1970s. And NIH caught up in 2017 with quite a very good paper in which they tried cannabis in many, many different seizure models. And they actually tried cannabidiol in many different seizure models and found that it worked on many different sorts of seizures. It's very broad spectrum. They also found that it was very non-toxic in terms of acute toxicity, at least.
Speaking of different groups doing research on epilepsy, could you tell us a bit about what Eplink is and what your role in it is?
Well, Eplink is the Epilepsy Research organization of the Ontario Brain Institute. Ontario is really quite lucky in that we have a virtual institute. They're interested in CNS disorders, and they have branches. They've got one working on depression, one working on cerebral palsy, on neurodevelopmental diseases, on neurodegenerative diseases. I think I said depression already. And ours is the group working on epilepsy. So we've got a little more than $1 million a year to spend, and it's spread across a bunch of different universities, different lecturers, but just everyone working to try to stop seizures and improving life for people that have them.